something like this. And there's this over here. So this is a benzene ring structure. And you've got that. And there's another nitrogen hanging off that. So you can see the R group. Anyway, so it's an amino acid derived signal. It's tryptophan. This is what the amino acid tryptophan has, this R group, and the C-terminal end was chopped off. And that's what gives you 5-hydroxytryptamine, 5-HT. It's kind of interesting. So it really is a derivative just of an amino acid. The other amino acid that's used is glutamic acid or glutamate. What's the difference between glutamate and glutamic acid? What's the difference between hyaluronan and hyaluronic acid?
One's more basic and one's more acid.
What's that?
One's more basic.
More basic? Okay, why is it more basic?
Does it have to do with the presence of the hydrogen?
Yeah, it's just, it's kind of a way of, it's the difference between the acid and the salt. You ever heard about this before? Comes up in chemistry quite a bit, in inorganic chemistry. So, in biology, we are involved with chemistry, but we don't, we're not real rigorous on the chemistry, but we use it as a tool to understand biology. And we are made out of chemicals, so, this is important. So, yeah, if you have a sodium ion present, it will neutralize the acid, and so then you have the salt. And if there's no sodium ion present and it's in solution, it can have, most of these things are organic acids, so they're COOH, and so you have the acid form. So these neurotransmitters, you have glutamate, serotonin was derived from tryptamine, aminobutyrate, GABA, and so yeah, there's also an OH here, which is so the reason to even talk about those guys is because glutamate is the most common neurotransmitter, and for an excitatory transmission. And GABA, a derivative of that, is the most common inhibitor. So, you can see between the serotonin and glutamate and GABA, GABA is inhibitory. And the glutamate, we're saying that one's stimulatory. And these are the most common ones. So suddenly, oh yeah, that's worth knowing. Amino acid derived neurotransmitters. And then they have this huge effect. So if you block serotonin, actually what you do is you change the way it's metabolized. There's a select, in the book they talk about select serotonin uptake inhibitors. So it's the way that the amount of serotonin that's taken up by the receiving neuron is affected by your drug. And then, what effect does that have on a person? Because you're talking about electrical impulses, and we've got billions of neurons, and then you make this drug that changes the way that the serotonin's taken up, and then you get effects on, reducing depression or anxiety, and much more complex mood behaviors, aggression, and all these other things. So it's kind of interesting that you could work at such a crude level, but there's going to be side effects. So obviously that's something from. So so enough of that. So we talked about the glial cells being a mesenchyme-derived cell, but it doesn't produce a lot of collagen or extracellular matrix, but it makes a lot of processes that interact with neuronal cells and helps to support neuronal cells. But that overall thing, did we mention this term, the neural pill? Just want to make sure we got that in there. So the neural pill is that mass of all sorts of branch structures, and within that you see cells, but there's a lot of fibrous looking material, and they are actually axons and dendrites, and everything is really dense in here, but it's the neural pill. So when we show a picture of that and we ask you to identify, well, what's in this area here, there'll be an arrow pointing to, well, neuron, maybe some glial cells, and then the general area where it looks like a lot of filaments, that's the neural pill, and it's not connective tissue. So it's not connective tissue, it's not extracellular matrix. And so on the next exam, when you get a picture, it sort of looks like connective tissues. Make sure that if it's in the section with other neural stuff, it's probably the neural pill. That's probably the correct answer. Okay, now we have the chance to talk about a couple of other cell types. So that's good, because this is a histology class. We even got into chemistry here for a moment. So the glial cells. So there are neurons and then glial cells, and everything else was a glial cell. And so in the neural pill, well, this complexity, the things that look so fibrous, those are actually parts of cells. And there's a couple of different types of glial cells, and so we should know what those are. Oligodendrocytes. Let's put that one up there. So a site is a cell, right? So we're comfortable with that. Cell, site. Oligodendrocyte, so dendritic. Oligo, multimers. So the oligodendrocytes, they make myelin sheaths. And the myelin sheet is what the Schwann cell is making, but there are other cells that make myelin sheaths, and the oligodendrocyte is one. And we were saying that the myelin sheaths contain a lot of lipid, and they are insulators. But not for an electrical impulse exactly. They maintain the ionic concentration necessary for the neuron to transmit its signal. Sorry about this, but over in my notes here I have just one more thing on the glutamate. So I started drawing the serotonin and the tryptophan. Should have drawn this in a different area, but so glutamic acid. Hey, by the way, little factoids that help biologists to understand chemistry. So, we were saying about glutamate versus glutamic acid, hyaluron-N versus hyaluronic acid. One of the things about just structures like this, so it's organic chemistry, right? Every time you draw a line like that, any kind of change in direction of the line, what does that represent? What's happening? What molecule is here? Carbon, right? So that's key to know. It's always carbon. So if you draw a structure like this, what's at that point? Carbon. What about over here? It's carbon. Yeah. Carbon, 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 carbon. Right? You guys know that, right? So that helps you kind of understand what things really look like. So rather than drawing carbons everywhere, they just draw these lines and they shape them different ways, and it kind of reflects the stereochemistry sometimes, whether something's L or R or whatever, and they'll draw them certain ways. Also, if there's going to be a cyclization of the carbon ring at some point, they'll start to draw it as a partial circular looking thing, but every time they draw a bend, that's just a carbon. They could draw it out and just do, carbon, 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 but that seems unnecessary to do that. Rather than drawing out this linear molecule, they start to fold them and everything. So that was glutamic acid, and then GABA just, in this diagram of that, it's just, if we break this chain up and we just draw it more linearly, you can see where that is. It's basically the same thing. Anyway, I just wanted to mention that. So these glutamic acid so the main stimulatory synaptic chemical is very closely related to the main inhibitory synaptic chemical. And what are we talking about when we're talking about chemicals? We're talking about small organic molecules. They were derivatives of amino acids. Okay, back to cells. So the astrocyte. And astrocytes are really interesting, I think. So they're in the central nervous system only, so they're not in the peripheral nervous system. And it's the most common glial cell. So, this is a typical test question too. Here's a bunch of cell types. Which ones are glial cells, which ones are neurons. Astrocyte is the most common glial cell and a member of the central nervous system, but not the peripheral nervous system at all. These are so common that they came up with a categorization of the astrocytes. There's the fibrous astrocyte, and it has fewer processes, but they're longer. And then there's the protoplasmic. So these are types of astrocytes. A fibrous astrocyte versus a protoplasmic astrocyte. These are shorter. They have shorter branches. And why even discuss these at all? Well, this is very common in gray matter of the brain. Actually, I think what I'm going to do for the next class, for Wednesday, since I have kind of a problem with this DVD thing, I'll just download pictures and I'll show a bunch of pictures so that you can, we can spend a good amount of time sort of looking at, well, what does a slice of gray matter look like? What does a slice of white matter in the central nervous system look like? And then what do these different cells look like? So there's the fibrous astrocytes and protoplasmic astrocytes, much more common in gray matter in the brain. So we've discussed the oligodendrocyte as being a type of glial cell that provides the myelin sheath insulators, and then this is another glial cell, an astrocyte. Doesn't look anything like an oligodendrocyte. The oligodendrocytes are actually wrapping around the axons, and they have a bunch of filamentous sort of processes that come out, and they wrap around axons. Whereas the astrocytes, they can connect a lot to blood vessels. They're these funny-looking cells that have these arms that come out, and they have like these feet on them or whatever, and they. So if this was a capillary down here, so capillary is going to be shuttling around red blood cells, which are known as erythrocytes. Boy, this selection of pens is pretty bad. Sorry about that. Get more pens. Okay, so that's supposed to be red. So it's binding to a capillary. So why is that useful? Well, the astrocyte can help shuttle nutrition to the neuron. It's kind of an intermediate. So it's a glial cell that can take support from the vasculature and soak up some nutrients, even help some of the gas exchange, and then interact with neurons and help feed it. So it's kind of a hand-to-mouth kind of situation with astrocytes feeding neurons. And so that's why they're so important. That's why they're the most common glial cell. Food is always a big deal in biology. Right, you got to eat. And so there's lots of these protoplasmic, shorter branched astrocytes in the gray matter of the brain. Brain uses up a lot of energy, actually. There are other cell types. So there's epidermal cells. So these guys are kind of epithelial looking. They had a picture of like a duct looking thing, and then there were these looked like a layer of simple epithelia all the way around. And it would be easy to guess that that is an epithelia, but it's actually epidermal cells. So what was different about these? Well, these are, I think they're little cilia, for one. But they could still be a ciliated epithelia. The other thing about them is they don't have a basement membrane zone. So there's no BMZ or basal lamina. And then they don't actually even flatten out. You know how epithelial cells, when they're cuboidal, they're kind of, cobblestone looking, and they kind of connect with each other like that. So this is where the basement membrane zone is. That's what epithelial cells look like normally. But these epidermal cells, they don't have a basement membrane, and actually they're not even flat on the bottom. They kind of just go into the surrounding tissue like that. Isn't that funny? It's weird. So that's what the epidermal cells are, kind of inspired by epithelial cells, but they're missing some key things, like the basement membrane zone, and the fact that they're rather than forming a flat surface to interact with mesenchyme, they go into the surrounding tissue with these processes here, and then they're ciliated. So why do you need these? Well, you need to shuttle around the spinal fluid. So cerebral spinal fluid. So let's have some ideas from you guys on what's the use of the fluid there, cerebral spinal fluid.
It accessed the shock absorber.
Okay. Shock absorber. Your brain is sitting in your skull, and it would. What's to stop the brain from hitting into the skull? I mean, it's a big piece of bone. It's a cavity where your brain is sitting in there. Is your brain? How is your brain situated in there? Is it right against the skull? No, it's actually floating. Have you ever seen Futurama?
Yeah.
So in Futurama, the cartoon thing, the guys in the future, there's a couple of people with these glass bowls on their heads, and so their brains are floating in there, I think, right? Is that the way it works? Yeah? Or is their whole head in there? Just the brain? Yeah. But that's actually, kind of realistic, because that's what our brains are floating inside a bunch of fluid. So the cerebral spinal fluid is suspending the brain. So the brain is kind of heavy. It weighs a couple pounds. And if there wasn't any fluid there, it would be sitting there like, this thing on a desk. I mean, there would be friction. It would be weight, right? And your brain can't be crushed. It can't have the weight. It can't support its own weight. It needs to be floating in fluid. So the brain is actually floating in the cerebral spinal fluid inside of your head. So, and then these epididymal cells, they're controlling the flow of cerebral spinal fluid. That's a pretty darn important job. And then there's the structure of that, and they don't show you a cross section of the brain, so then it gets kind of weird. But there's this ventricular system. Well, we've talked about the circulatory system. We know that there are veins and arteries, and we talked about the lymphoid system too. So there's fluid there, and then there's also interstitial fluid. There's fluid inside the tissues as well that's related to the blood. But there's this ventricular system inside of your head. So your brain is sitting there, and we said it's floating. Your brain is floating inside of cerebral spinal fluid. And then there's the ventricular system is a set of 4 cavities inside of the brain itself, kind of spread out like that. And, I'm drawing it 2D, but it's actually fluid that goes inside the brain. The brain is not just a solid chunk. If you looked at it, you see slices, and it's this convoluted thing, and then towards the base, you see that there are these. Oh, there's this huge cutout. What is that huge cutout? Huge cutouts are the ventricular system, a series of 4 major canals inside of your brain, and there's cerebral spinal fluid in there. And so, not only is it cushioning, but it supplies some nutrition as well, or some ions anyway. And so there's these epididymal cells that we're interested in that look like epithelial cells. They're not quite epithelia because they don't have some of the key things, but these cilia help shuttle along this spinal fluid, and they actually help make the spinal fluid as well, which is kind of a big deal. So there's the ventricle lining, which is an epithelial-like lining, and that lining is what's helping to produce the cerebral spinal fluid. That's pretty important. Reminds me of a duct. We didn't really go into ducts as much as we might have, but epithelial cells are lining these ducts, and then they produce these substances that are secreted. And so these epidermal cells are producing cerebral spinal fluid, and they're producing it into this ventricular system. Okay, there's a couple of other things. So, yeah, we don't have pictures of the brain. That really bothers me. Maybe we could get one online. I'll try. You can see in a course like this, so this is histology, but in order to really understand what tissues look like, you'd have to understand what the anatomy is. It's partly anatomy. So let's just see if we can get a picture. No, there's lots of pictures. Let's put some up. Okay, so this is not a course in anatomy or even physiology, but it is histology. So we're interested in, what would things look like if we take different slices. For us, we're going to focus just on the cerebrum, and so for us, we're kind of just thinking there's a cerebrum and a cerebellum, and there's white matter and gray matter. If we look at, there's a picture of what your brain looks like. There's two spheres, the hemispheres of the brain, left and right hemisphere of the brain. Left. If we were just brains, we would look like this poor guy here. Eyeballs and brains. Yeah, here we go. So the cerebrum includes, frontal lobe, parietal lobes, occipital lobe, temporal lobe. But I'm not going to ask you anything about the lobes of the brain. There's just the cerebrum and the cerebellum, and a brain stem that goes into the spinal cord. And I wonder if they have the ventricular system on here. Let's see, yeah. Yeah. So we don't actually discuss any of this in the class, but there was, let's see, besides the meninges, the dura mater, the arachnoid, and the pia mater. So you need to protect the central nervous system, and you're going to do that with some membranes. And those membranes are called meninges. And I don't see any of these pictures that don't show meninges. A lot of silly pictures on this. Let's go back. So here's your brain, and there's white matter and gray matter, and there's a space between your brain and the skull, and that's going to have cerebral spinal fluid in it. There are membranes. There's a dura, arachnoid, and pia mater brain. So the pia mater is right next to your brain, so it's the softest membrane, as you would expect, because it's right on top of the brain tissue itself. The arachnoid is part of this big space here, which has got cerebral spinal fluid in it. It's the membrane sort of defining that space on the outside. So the pia mater is the membrane on the inside, right next to the brain. The arachnoid is the membrane that's outside of that, but in between there's lots of cerebral spinal fluid. And then the dura mater is a very tough membrane, which you'd expect. I mean, so that's the one that's actually right next to the skull. And that can be the thickest membranous material because it has to grind against the skull. And if there's some damage, if you're hit in the head or whatever, the dura mater, you want that to be pretty tough so that there's not going to be physical damage to the brain itself. You can see some care was taken to try to protect our brains. Let's see some different pictures of that. So this is again under the category of meninges, right? So if you're asked about the meninges, there's three that I want you to know: the dura mater on the outside, the arachnoid that's in between, and it does contain blood vessels, but filled with cerebral spinal fluid, and then the pia mater, which covers the brain itself. So that's the brain, and then okay, the blood-brain barrier. BBB. It's made up of. I always thought that it was a really well-defined, simple membrane like what I'm used to seeing with the skin. But it's made up of. The main structural component is capillary endothelium, and they're tightly sealed together. But there's also this perivascular astrocytic feet that are part of that, and they envelop the basal lamina and the capillaries. Remember we were talking, when we draw the astrocytes, we had the astrocytes with their little feet on the capillaries, and so the blood-brain barrier involves a lot of astrocytes, and they help define the properties of the barrier, but the main structural component is this capillary endothelia. So what is the endothelia again? What does that look like, an endothelia? Came up before, we talked about endothelia. They're kind of epithelial-like, endothelia. Why would they be epithelial-like? Where do we come across the endothelia again? Yeah, inside of the capillary, so it was the lining of blood vessels. And so the endothelia was lining all the blood vessels. All the different blood vessels we talked about, they all had endothelial cells lining on the inside. And then we were saying that the blood that's pulsating through our vascular system is, it's the outside world. I mean, as compared to what's inside. Compared to packed tissue of the body, the blood vessel has this blood. Some people think that the chemistry of our blood was designed from seawater. If you look at the ion content of blood, that it has some similarity to ocean water. It sounds kind of interesting. So then, the idea is that as more complex organisms arose, that they were probably just squishing some seawater through to make sure all the little cells in the colony were seeing water. And then if that closes off, well, then you need some dedicated seawater to circulate around all of the cells. And so the endothelia is lining these vessels, and it's against the seawater, so it's the outside world. So anyway, so the blood-brain barrier is made up of capillary endothelial cells. Why is the blood-brain barrier such a big deal? What happens if you get, well, it seems like our central nervous system is sealed off, and much better than the rest of the body. So that makes some sense. Why do you think you'd need to do that, Dave? Why do you want to seal off the central nervous system?
Because if blood is toxic, and if it touches an organ, it's just gonna kill it. Or if it touches the brain, then it'll destroy some of the matter.
Okay, so you don't want bleeding in the brain. You really want to carefully control chemicals too. The neurons are really affected by things circulating. So you want to control the chemistry a little bit better. But if you got a big concentration of neurons, you want to control the ion concentrations very significantly. That's why we've got all these glial cells, we said, right? We've got 10 times more glial cells than we have neurons in our brains. Why doesn't anybody talk about the glial cells? It's always neurons. Well, they get top billing because they're doing kind of the critical work, I guess, but the glial cells support them. And so they protect them, support them, but they maintain a very narrow chemistry with the ion concentrations and allow those electrical impulses to be transmitted effectively. And also, so I think that's one reason to have a blood-brain barrier. But what about infection-wise? Right? I mean, it's one thing to get an infected finger and another thing to have an infected brain. If you lose your cognitive ability, you're going to be in bad shape. You're going to be making bad choices. And so, I mean, maybe you won't be able to get out of bed, at that point. I mean, if something goes wrong with your brain, you're in big trouble right away. So having a blood-brain barrier is a nice protective thing. What do you do? Is there any protection for the brain as far as infection? So the rest of the body, we talked a lot about leukocytes and lymphocytes, the adaptive and non-adaptive immune system in our bodies that maintain things. But what about the brain? What's the deal there? We have leukocytes and lymphocytes in the brain?
Not specifically microglia.
Yeah, there we go. So we have the microglia in there, and that replaces these other cell types. So the microglia look like neurons. Figure 912. This guy here sort of looks like a neuron. It looks like a neuron that doesn't have an axon, an anaxonic neuron. Right? So there's no long process with an axon. It just looks like a bunch of dendritic processes around this. And it looks very nerve-like. But is it a nerve cell? Not really. It's a glial cell. So that's kind of a trick question there. I mean, what the microglia, what's its function versus what it looks like? And so I think that's kind of a fun question, something that you should know. So it has this immune function. It's actually searching for possible contaminants, viruses or bacteria or whatever shouldn't be there. So and it's derived from what kind of cell? So the cell that does a lot of chasing around inside the tissues.
Macrophage.
Yeah, macrophage. Where does the macrophage come from?
Megakaryotes.
Yeah, it comes from monocytes. Monocytes give rise to macrophages, and so in the brain, the microglial cell is derived from macrophages, and yet because it's hanging out with a lot of neurons, it ends up looking pretty neuronal-like. but when it detects something, all those processes that it has that look like dendrocytes, they compact down, and so it can look a lot more circular. But it's so it's the neuronal cell that it's the non-neuronal cell that looks like a neuronal cell, and its function is to take care of possible contamination. Okay, I think that's pretty good. We're kind of out of time. Peripheral nervous system, yeah, there's some things to know about that. Might want to look at that part. We didn't really get into that. But that's, the afferent versus efferent. So afferent signals coming in, so they have to come in through the peripheral nervous system. And it's a very similar situation overall. Although you've got these groupings of nerves, which typically have some blood vessels. There's in figure 926, peripheral nerve connective tissue. Just looks like this. We've got, as part of this whole assembly, you've got some, blood vessels coming in and supplying nutrients, and then you've got a whole cluster, bundles of axons covered with neural sheets. And then that's where you get the endoneurium, perineurium, and epineurium. So just those three terms, and that's part of the peripheral nervous system, what slices look like. Okay, so on Wednesday, I'll have a lot more pictures, so we'll go through just pictures. So we'll start out by flashing some pictures of nervous system stuff and identifying that. And I'll give you some review sheets, some questions that might be on exam 3, so you can study from them. Yeah. I was thinking Monday. What do you think? Yeah. That sound good? Okay. Yeah. Better Monday than Wednesday.